Thank you very much. It's really a pleasure to be here. This is the first time I've ever been to a conference on either fascia or acupuncture, so if something's not clear, just interrupt. Okay. I'm really a basic scientist. I never worked with a human ever, except we are collecting lymph from melanoma. Sorry, there's feedback. If I get too close, I don't know why. But anyway, all right. My lab works in the lymphatic system, and we've been studying it from every different angle for at least 20-some years. And today I'm going to try to tell you some new things that are coming out in not only just my own lab, but in general in how we're understanding the lymphatic system. And it's so much more than just a transport system. And I think it's really relevant to this community, and I'm really happy that I came to this meeting, so thank you again. All right, so for my disclosure, as you all know, lymph node involvement is a primary prognostic indicator of cancer. If there's metastasis in the lymph nodes, then it's probably going to spread into other parts of the body as well. But what is not known still is why. Why is lymphatic involvement promoting metastasis? One might think, oh, well, if there's more lymphatics there, tumors can escape more easily. But, lymphatics are there to begin with. Why do they need to expand? Furthermore, lymphatics lead to lymph nodes, which are the sites of primary adaptive immune responses. In fact, there were some very interesting studies done where a very invasive mouse melanoma was just injected into the lymph node of a naive mouse, and it was rapidly killed by the immune cells. If you instead inject that exact same tumor into a tumor-draining lymph node of a mouse that bared that tumor but hadn't metastasized, those cells just thrive and survive. So the lymphatics are actually doing something to change the tumor microenvironment, and that's where we've been looking mostly in the last, especially decade of the work we've done. So I hope to convince you that they're not just passive transporters. And by passive, I don't mean active pumping. I mean that transport is passive in that when they pump, they generate a pressure gradient, and still that's considered a physical passive process. But actually, they do a lot to regulate a lot of things. So we think about them, and this was a movie, but I'm sorry it's not working. But we think about them as transporters. They transport fluid. If they don't work, you swell. They transport cells to the lymph node. They transport antigens. I just showed this also to say, in this movie, which is just a picture now, there are green leukocytes going through this blood vessel. This is the lymphatic vessel here. This is just immunostained in a live ear tissue for a protein that just binds to to basement membrane, and so you can kind of see everything. You can see the fat cells, you can see muscle, nerves. But what's really nice is you can see this is also stained for a chemokine that I'm going to talk about in my talk called CCL21. This is a lymphoid homing chemokine, and these leukocytes will exit the blood and find this patch of extracellular CCL21 and enter the lymphatic there. So CCL21 is really critical for immune cell entry. Oh, this movie works, good. Okay, these are tumor cells moving into the green are melanoma cells. Oh, sorry. Yeah, you're right, they're both green. The movie doesn't work. Sorry. Anyway, let me back up one more time. Well, it should just play naturally, so let me just see. Oh, there. Okay, so these are melanoma cells, and they will interact with this lymphatic vessel. It's more, sorry you can't see the lymphatic vessel very well, it's red. And what's interesting is you see those cells moving in, like one cell will enter and then a whole bunch of cells will follow in that same hole. But once they're in, they don't necessarily just flow to the lymph node. They stay there, they hang out, they interact with that lymphatic vessel, and they're actually, we now know, manipulating that lymphatic vessel. And I'll tell you why in a second. But they really exchange information with the lymphatic endothelial cells. And, they train the lymphatics to kind of help protect them against immune responses. Okay, so again, as I mentioned, we think of the lymphatics as the escape route for cancer, but they go to lymph nodes. That's like saying, like, escape route from a prison is going to the police station. So, and the lymph node is where adaptive immune responses take place. And so these things are very well coupled. Sorry, I don't. This CCL21 chemokine attracts immune cells. When you get inflammation, this is upregulated. Immune cells like dendritic cells will enter into the lymphatic vessels more readily. More fluid also goes to the lymph node when there's inflammation. And so as there's more fluid, more interstitial flow, and more trafficking and transport to the lymph node. Now, from an immunologist's point of view, the lymph node really is just a bunch of cells interacting and driving an immune response. But we also have to remember that there's a lot of stuff in lymph, and it's not just extracellular fluid that leaked out of the plasma. It's basically information rich. It has all the information of the tissue it drains. And you have almost 1,000 lymph nodes in your body, and each cluster of those drains one particular tissue and contains all the information from that tissue. And so this tissue, it's not just, it's the proteins that are in the extracellular matrix. There are things called antigens. These are things that the immune system recognizes. So when you do have an inflammatory event or even a pathology, you have danger signals. Like an infection, I'm sorry. You have danger signals, but then you also have a lot of your own proteins that are breakdown products or from apoptotic cells. And so lymph is really full of these antigens. And what we're finding is really that the lymphatics and the lymph flow to the lymph node really helps maintain tolerance to those self-antigens. And we have done some studies, I'm not going to show you the data on here, but we've done studies where we have mice that don't have lymphatics in their skin, and they are very prone to develop autoimmunity in the skin-related autoimmunity. It's because they're not sampling and constantly being bathed in the self-antigens. And so this is a really important part also, I think, of normal lymph flow. And I think it's probably also, I haven't seen any clinical studies to correlate whether people with lymphedema are more likely to get autoimmunity, but I would suspect it's probably the case. That you just don't have this natural, your body needs to maintain constantly tolerance to self-antigens. Okay, in the lymph node, of course, are where these immune cells interact. The information is carried there, and that's where the information is mounted. So if I were to give you, one more thing is that lymphangiogenesis is when lymphatics expand, as already mentioned. But it occurs in all types of inflammation. It occurs in lymph nodes, even after a vaccine. It's also in tumor-draining lymph nodes. The red here are lymphatic vessels. We see it in pulmonary infection, even in transplantation. And we still don't really know why. We don't know what role that plays. So these are the questions. What is the function of an expanded lymphatic network that we've been asking? I really believe in this meeting is great because it's all about integration of various perspectives. And I think with the lymphatic system, it's not simply a transport system, but it's really an integrated part of all your organ systems. It's part of the circulation. It's part of the metabolism. It's where all lipids are absorbed from your diet. It's part of the immune system. It's even part of your salt balance, and people are finding now that in skin, for example, salt is regulated in skin actively by lymphatic vessels. So it really helps maintain homeostasis, and I think that's why we haven't had a lot of research to date compared to how much we should be having on understanding lymphatic biology. It's because it's not so obvious when the lymphatics don't work. If their goal is really to maintain homeostasis and help everything run smoothly, these problems manifest slowly over years. People even develop lymphedema slowly. And so you wouldn't necessarily, know that that was due to a problem in lymphatics, or if there's, you can develop like autoimmunity and have a lot of health problems as you age, you wouldn't necessarily think that was a lymphatic problem. But I think the lymphatics could be very critical to a lot of those things. Okay, so I'm just going to give a very simple analogy on immune surveillance because I'm suspecting that not everybody here knows a lot of immunology, or, I didn't until I started working in this area. We've got patrol cars, which are your dendritic cells. They basically are assigned to a tissue. They just constantly sample. They're constantly going around. They're picking up things both safe and danger and giving all that information to the lymphatic system and to the lymph nodes. And we now know that they don't even need to get to the lymph nodes to get the information there. They can secrete something called exosomes, which are little vesicles that they pop out, which contains information, microRNAs and proteins, antigens, that flow directly to the lymph node. So they communicate that information. And this doesn't necessarily have to be danger like pathogens. It could even be apoptotic cells. It can be, they need to check, just like a patrol car, they need to just check what's going on. They need to find anything that's wrong. If it's, a fire hydrant is leaking, they need to call that in, or if there's a terrorist, they need to call that in. So it could be something pretty simple, like cat stuck in a tree, or it can be something really serious. So the communication center, the lymph node, is like the headquarters of the police, and information is given, like what the person is wearing, where he was last seen, what kind of car he was driving, like what to look for. That's the information that's given to this guy. And then he has to go and dispatch the appropriate response. And the appropriate response depends on the danger. So with the cat, it's guy in the ladder, a fireman, and with that, it's the SWAT team, and there's everything in between, right? So this shows you the dendritic cell really exchanging that kind of information with the T cell. So the dendritic cell is the one that picks all that stuff up, and then it goes and activates this T cell. And so it gives it this specific information through these receptors, but it also secretes various cytokines to tell it what to do with that information. And we're finding now that tolerance maintenance is actually much more important and regulated. There are more cell types involved in, for example, than inducing an immune response. So, I'm just saying that both have to be considered, and we often think of the immune response as being the pro-inflammatory, the activating a T cell to go kill, but we also have to tell T cells who to ignore. So maybe a better analogy would have been like an airport security check, where everybody's going through, you're assumed safe unless you have something bad in your luggage, and if you're already detected to be a frequent traveler that pretty much is not on a terrorist watch list, you go through the TSA pre-check line, which is like, it's kind of like, that there's the same thing in the immune system where T cells are told, if you see this thing, just ignore it, it's safe. Okay, and this is really some of the mechanisms by which cancer cells escape immune detection. So the immune system can detect cancer. It's rapidly dividing cells. It's weird. It's not normal. And the immune system gets alerted. Often there's mutations that they can recognize. And in fact, most of you who do not have cancer have a good immune response to any cells that come up and become cancerous. They're killed rapidly by the immune system, and you never get cancer. But the ones that do grow and survive escape that immune detection, and they can do so in three ways: inducing ignorance, inducing suppression, or inducing tolerance. Ignorance, the immune system doesn't even know it's there, it's hiding. Suppression, it's dampened. And then tolerance is like turning the T cells into zombies. Yeah, really. Okay. So one of the things my lab has really been trying to understand is maybe we should think about the lymphatics in cancer as really helping to avoid immune detection, because the lymphatics are the ones that connect it with this lymph node. And so more lymphatic involvement, the more information is communicated to the lymph node. What we found, I mentioned, is that lymphatic drainage is critical for immune recognition and tolerance. Local lymphatics scavenge abundant antigens, and we found that they can also transmit that information directly to T cells. So they pick up the exosomes that are secreted by dendritic cells, and they can present that information themselves to the T cells as well. And that's, again, for promoting tolerance. And importantly, the sentinel draining lymph node, often taken out by the surgeon, contains all of the information about the tumor, all kinds of things, what protein, what is its barcode, what is its fingerprint. But they're all immune suppressed and tolerized, as I mentioned. I know I've said this like many times, but it's a really critical and kind of, it's kind of a new way to think about this stuff in how we understand lymphatics in cancer. So lymphatics are basically transmitting this tumor-specific information and telling it to be safe, that it's safe, but it's deceptive. All right, so one thing we've done to try to take advantage of that is to say, well, rather than take out the draining lymph node, why don't we just turn that into a vaccine? It has all the information, but it has the wrong instructions about what to do with it. So we developed, so again, can we target this to make it reprogrammed? So we developed, in collaboration with Jeff Hubbell's lab, who is a biomaterials scientist, nanoparticles that are so small that when you inject them into the skin, they readily flow into lymphatics. And these are really nice to target things to the lymph node. They're big enough where they kind of get retained in the lymph node, but they're small enough to readily get there. And so we could deliver danger signals. And I want to show you just this piece of data. This is in a very aggressive mouse tumor called a B16 melanoma. And this white bar here is just the tumor growth over time in an untreated mouse. Here is this light blue bar is if we vaccinate, and sorry, this should have been on here, but we vaccinate these mice at day 7. So right here, once the tumor is getting into an aggressive growth phase, we vaccinate. And if we vaccinate in a non-draining lymph node, so the opposite, contralateral, we see that the vaccine, so we vaccinate at day 7, you see a response starting at day 14. So that means it takes time for the T cells to sort of get, proliferating and start killing the tumor, and it starts to regress. But because of that time delay, the tumor can meanwhile mutate and find other ways to escape as it's starting to get slowly killed. And it can respond, it can grow, it becomes completely ignorant of that immune response because of the mutations. So it relapses. Whereas if you instead target the tumor-draining lymph node, we see, first of all, it's much quicker when it starts to work because the T cells are already there, they're already educated. They just need to be told what to do. They need to be just turned in the other direction. And they rapidly respond. The killing is quick, and it's complete, and they never, ever grow back. And we've let these go for a very long time. You can see 100% survival versus these 2 survival curves. So that's like one way we can think about how we can use knowledge about lymphatics and how they're promoting the cancer to actually use it to our advantage in treating cancer. But the bigger question is really like, why do tumors induce lymphangiogenesis? So all of what I told you, that should just happen with a normal lymphatic network. Why do they need to expand? Why does it expand in all types of inflammation? What do they do to the microenvironment? So in this second part, which is shorter, I'm just going to tell you about how the lymphatic activation is altering the stroma and the microenvironment. And I think this is not only happening in cancer, but in any kind of chronic inflammation. And we're studying this also in other models. So first, a human genome database. We can look at melanoma patients. We can make a lymphatic score that is just some genes that are related to lymphatic expansion. And we see a direct correlation between lymphatic involvement in the tumor and immune cell infiltration. So the more lymphatics, the blue are low, these are mid, and the yellow are high. Sorry, the opposite of what I said, sorry. Yellow is low and blue is high. So these are just different types of immune cells that we look for genes that represent things like macrophages, dendritic cells, and natural killer cells, T cells, whatever. And in all cases, there's a lot more when there's lymphangiogenesis. So lymphangiogenic tumors attract more immune cells. Why is that? Well, go back to this molecule we've been studying a lot, which is CCL21. And that ends up being one of the most highly correlated genes with VEGF-C, again, in these 266 patient samples. This is just direct VEGF-C expression versus CCL21 expression, and it's a very strong correlation. So that means this is kind of one of the things that we were kind of excited to see, because we had been publishing for a while, like papers showing that VEGF-C does upregulate CCL21, not only by lymphatics, but also by other stromal cells in the tumor microenvironment. In fact, here's an image showing CCL21 expression in green. Live1 is a marker for lymphatics, so this is the tumor. And here's one that's overexpressing VEGF-C, this lymphatic growth factor, and you can see way more CCL21, not just by the lymphatics, they're upregulating CCL21, but also by the fibroblasts in the stroma. So it also is upregulated in the draining lymph node, but not the non-draining lymph node. CCL21, as I mentioned, it's a lymphoid homing chemokine, but it attracts certain types of cells. It attracts naive T cells, regulatory T cells, or Tregs, dendritic cells, and lymphoid tissue inducer cells, which come in and transform the tumor microenvironment. And so we think that in this paper that we published several years ago, we showed that when these lymphangiogenic tumors that express high levels of CCL21, when they have CCL21 knocked down, they lose these stromal changes. And the stromal changes are these are a lot of markers here on the left that are just typically found in a lymph node. And these are expressed by lymph node stromal fibroblasts. And these are more like myofibroblasts, really. They're really they're contractile. They have high levels of proteins that are like myofibroblasts-like. And they are completely in tumors that are lymphangiogenic and CCL21 high in the stroma, and not when it's knocked down. So why is that interesting? Well, tumor-associated fibroblasts are, as I mentioned, very similar to lymph node stromal fibroblasts, or FRCs. And these FRCs, their job in the lymph node is to make a good matrix, to keep bundled collagen fibers, conduits, to allow and secrete a lot of proteins that allow T cells and dendritic cells to interact so that they can get activated. So this is a prime environment for immune cell activation. Now, the other thing that VEGF-C was correlated with in these melanoma patients was TGF-beta. And this was strong. It was so strong, we were super interested in this. And so we looked in other cancers, and here's just an example, breast cancer, very strongly correlated as well. Now, TGF-beta I bring in because it's, again, this is what's actually driving the stromal remodeling. TGF-beta is what makes fibroblasts into myofibroblasts, and this is what drives, this is what drives their contraction and their matrix production and all the things that we see that happen to cancer-associated fibroblasts, but also fibroblasts in wound healing. So when you have TGF-beta rich environments, they're more stiff, there's more matrix proteins, there's more contraction. And this has been correlated, interestingly, and you'll hear later this afternoon, this is really correlated with invasiveness in breast cancer and bad prognosis. And so it's very interesting that the lymphatics, which are also correlated with bad prognosis, are a main source, we think, of TGF-beta in the tumor microenvironment. So I wanted to show you, this is a mouse model of, it's actually a genetically engineered mouse model, so they spontaneously make breast tumors. And this is a mammary fat pad, here. This is the tumor, this is the draining lymph node. Actually, there's a lot of tumor cells, tumor nodules coming up, but this is the main tumor. And so if we look at this lymphatic vessel in pink, that's the lymphatic marker. And then if you look in blue, that's TGF-beta. there's the overlay. So white means that the lymphatic is secreting TGF-beta. Look in the lymph node. This is now the whole lymph node. These are the lymphatic vessels in the lymph node. You see the TGF-beta is strongly correlated. This was fascinating to us because it wasn't known that lymphatics could be a main source of TGF-beta. So we took the lymphatics out of this mouse model, and then we ran flow cytometry to see, is it really in the lymphatics? Are they actually making it? And the answer is yes. In these tumor-bearing mice, the lymphatic endothelial cells, and there is high levels of VEGF-C in this, they have a lot of TGF-beta. In vitro, we can culture lymphatic endothelial cells, throw VEGF-C on it, and you can see in a dose-dependent way, TGF-beta is upregulated. In vitro, we can do similar types of things. We can look at co-cultures between tumor cells, fibroblasts, lymphatic endothelial cells. We always culture these under flow, by the way, to maintain a normal microenvironment. You see that when these cells that are activating the lymphatics are present, the lymphatics are secreting TGF-beta. This is also here the matrix now. You can see a lot of matrix remodeling, stiffening, everything like that when the lymphatics are present. So if we go back to that mouse model, and now we block lymphatic activation. So we block the receptor to VEGF-C. That's this. Everything goes down. That means that the VEGF-C induced TGF-beta is actually enough to remodel the matrix. So here's TGF-beta in the whole tumor microenvironment. Here's a normal, this is just a control antibody, and here's the blocking antibody. And you can see it's significantly decreased, just as much as if we block TGF-beta itself. If we look at the alpha smooth muscle actin positive fibroblasts, which are the contractile myofibroblasts, again, when we block VEGF receptor 3, that goes down. And if we look at collagen remodeling, and this you can use various dyes, this is called Sirius Red, you can see the collagen remodeling is vastly reduced when we block lymphangiogenesis. So what this means is that the lymphatics are really doing a lot more than we thought. They're not simply an escape route. They are changing the whole microenvironment. They attract immune cells. They attract lymphoid tissue inducer cells, which changes the stromal matrix and makes the fibroblast activated. So this is through CCL21. They secrete, now we know, TGF-beta. That's not quite published yet. Hopefully it will be soon. And this, in turn, activates fibroblast, which leads to all these changes in the matrix that are classical hallmarks of cancer. The interesting thing about TGF-beta is it is also part of the immune system because it's a main suppressor. It's an immune suppressive cytokine as well. And so when TGF-beta levels are high and T cells get activated in that presence, that's the signal to tell them it's safe. So TGF-beta plays these dual roles. They are the safe signal. I told you, like, the lymphatics are telling the lymph node to take all that information about the tumor and say it's safe. Well, TGF-beta is one of those signals. TGF-beta and IL-10 there too. So it's really interesting that this is all part of the whole big package. So when you have inflammation, you activate VEGF-C, it's usually macrophages that come in, drives lymphangiogenesis, and then all these things follow in chronic inflammation. The lymphangiogenic tumors are more suppressed, okay? They contain a lot more immune suppressive cells, and I only have 2 minutes, so I'm just about done. Here are regulatory T cells in the VEGF-C secreting tumor. There's double the numbers, myeloid-derived suppressor cells and suppressive macrophage phenotypes. And then finally, if we first vaccinate the mice against an antigen that we make the tumor cells express, this is a chicken antigen, so it's totally foreign. It's called ovalbumin. We first vaccinate the mice so they have a good, strong anti-ovalbumin immune response. They have lots of T cells that are specific for that protein. Then we inject the tumor. If it doesn't have VEGF-C, the vaccine sort of works. There's enough T cells to control tumor growth, at least partially. If we have VEGF-C, again, they're lymphangiogenic, they completely ignore the vaccine. If they were vaccinated or not, they grow exactly at the same rate. If we block VEGF-C activation, we block that. We restore the anti-tumor immunity. So again, we think that the lymphatics are bringing information about the tumor or whatever tissue it drains to the lymph node, telling the lymph node what's safe. It's modified by the tumor to be immune suppressed but antigen experienced. When the tumor or chronically inflamed site becomes lymphangiogenic, then there are other stromal changes that include changes that attract a lot of immune cells and that change the stroma. And again, I said that TGF-beta is an important immunosuppressor, and in this way, we think the stromal changes that occur in the tumor due to lymphatics are tightly coupled with immune regulation. And in this way, the tumor really is hijacking normal features of the lymphatic system, which are designed to prevent autoimmunity, to their advantage. So do we think this is the reason we have lymphangiogenesis? By studying the tumors, we can hypothesize that maybe this is the broader function of lymphatic expansion, to really just help the body by taking in constant, abundant antigens and presenting it to your immune system constantly, telling it what's safe, that this is helping us to prevent autoimmunity. And when the tumors start using the lymphatics in this way, then they can escape immunity. And the lymphangiogenic tumors are harder to kill with immunotherapy. So with that, I'd like to end. Thank the people who did the work. A lot of this work was done by Jenny Munson, who now has her own lab at University of Virginia, and Amanda Lund at Oregon Health State University. And she's just started her own lab as well as these postdocs and PhD students in my lab. So thank you for your attention.